This video is about the central dogma of molecular biology. The best way to use this video is to ask yourself, every time I type out a new statement, is it a surprise to you or is it something you knew? What I'm going to do is type a bunch of things out one statement at a time and build a case. I'm going to build everything you need to get the central dogma at the amount of detail I want you to have it, and at every step I want you to consider stopping and asking yourself, have I heard this before? Is this something I know? Or does this sound like it conflicts with something I know? If it conflicts with something you know, please ask about it on the Q&A or in class. Okay. So let's get into some of the key things to think about. Here are some facts that you need to know or believe or whatever. These are a big deal and you need to know that. Number one, proteins are very important. There are many different proteins in a cell and they have different functions, and making the correct protein at the correct time is the single most important thing any cell ever does. Let that sink in for a second if you weren't aware of it. It's the biggest job any cell has. It's how cells accomplish everything they do. They need to make the right protein at the right time. Think about what proteins can be. They can be enzymes. And if you don't remember what an enzyme is, pause this video and look it up because they are very important. They catalyze chemical reactions. Not just that. So if you remember what a catalyst is, a catalyst makes a chemical reaction go faster. But it's not just that. Most biological reactions inside cells, if they're not catalyzed, they are so slow, they essentially never happen. If they are catalyzed, they happen essentially instantly. So an enzyme makes something happen that would never otherwise happen. And so cells can perfectly control their metabolism and everything they need to do if they have the right enzymes made at the right time. So enzymes are very important. Structural proteins are a big deal. They hold things together and they make shapes. And those are really the biggest types of proteins you need to think about. Enzymes really is the biggest thing, but there are a lot of others. So for example, it took me a second to think of it, but like flagellin is a type of protein. That's what flagella are made of. So flagella are these long corkscrews made of protein. Those proteins in the corkscrew itself don't do anything except be that structure. But the molecular motor that makes it spin is made entirely of proteins, and they are all classified as enzymes because they do chemical reactions. So that's super cool. Other proteins might be receptors, cell surface receptors that detect things outside of the cell, right? They could be regulators that help the cell make decisions and then enact those decisions. That's what proteins can be. Any different thing you can think of in a cell, the cell might use a protein to do it. That's why we always remember that cells have proteins inside them. We have to think about how cells make proteins and why they make proteins. So for now, that's important. The next important thing is what proteins are made of. Proteins are, you tell me, what are proteins? Pause the video, tell me what proteins are, and then see if your answer is the same as mine. Proteins are chains of amino acids. And there are 20, roughly, amino acids that can be used to make a protein. So how long is a typical protein? Is it 5 amino acids long? Is it 1,000,000 amino acids long? A typical protein is a chain of 300 amino acids, and they vary a lot. So very small ones could be 50 long, 50 amino acids. Very big ones could be 2,000 amino acids, but just think 300 would be a good guess. So each amino acid has its own chemical properties. And so if the wrong amino acid is put in the wrong place in a protein, it will change the protein's properties. We can say in general, the order of amino acids, also known as the amino acid sequence of a protein, determines everything about that protein's behavior and character. So you pretty much just have to trust me on that. That cells have to make each amino acid chain in the right way. They have to add the amino acids in the right order. And this is where we get to the central dogma of molecular biology, because DNA primarily encodes the amino acid sequence of every protein. So your genome encodes all of your proteins. That's what it does. That's the primary thing your genome does. The primary thing DNA is for storing the information that is used to make the proteins. So DNA is a stable, long-term storage molecule. You can keep a sequence, you can keep information stored in the form of DNA for the entire lifetime of the cell, and it will be stable there. What is DNA made of? Well, DNA is a nucleotide, or I'm sorry, nucleic acid. A nucleic acid is a chain of nucleotides. So, okay, so if it's a chain of nucleotides, what are nucleotides? For our purposes right now, nucleotide is a molecule that can be represented by one letter. And in DNA, the letters are A for adenine, C for cytosine, G for guanine, and T for thymine. Those are the four nucleotides that make up DNA. So a DNA sequence is entirely made up of those four nucleotides. The order of nucleotides determines the order of amino acids in the protein. Okay, cool. We're going to need one more piece of this puzzle, and then we will draw out the whole central dogma. RNA is a nucleic acid. It is a chain of these nucleotides, A, C, G, and U. So instead of thymine, it has uracil. Otherwise, it's very similar to DNA. RNA is less stable than DNA. It is used as a temporary or short-term storage of information. Okay? So, a lot of this starts to make sense the more you know about cells and how they work. But basically, I told you before that making the right protein at the right time is very important. You don't want to make too much. You also want to make the right amount. You don't want to make too much of it. And so this whole system is set up to help cells make proteins at the right amount at the right time. Now, I'm only showing you a tiny part of what cells do and how they control this. The central dogma is kind of the backbone of this system, and it's the major parts of the system and the major tasks they do. We could look at any one task in a lot of detail, but here we won't. So, the central dogma is this. DNA stores information. Let's back up. In the form of genes. Each gene encodes a protein. the DNA sequence of a gene encodes the amino acid sequence of its protein. The first step of making a protein is making an RNA copy of the protein's gene. Okay, so the gene is the information needed to make the protein. It's stored as DNA. So the first step in making a protein is making an RNA copy. That process is called transcription. I'm going to make that bold because you definitely have to know it. Transcription is performed by an enzyme complex called RNA polymerase. We can unpack this name a little bit. Polymer. You can Google what is a polymer. Well, I can tell you, DNA is a polymer, RNA is a polymer, proteins are polymers. A polymer is a big molecule made by attaching lots of little molecules together. So if you have a chain of nucleotides, that is a polymer of nucleotides. And so a polymerase enzyme makes a polymer, and the RNA polymerase enzyme makes the polymer RNA. Cool. So once we have RNA, the RNA made for this purpose is called messenger RNA or messenger RNA or mRNA. The process of translation makes proteins based on the mRNA sequence. Okay, that's translation. A ribosome can read sequence of nucleotides in messenger RNA. The ribosome can then translate that as a sequence of amino acids in the new protein. So the ribosome is what does translation, and it makes an amino acid chain. You can look up videos on YouTube of how this happens, and you can see really cool videos of how this happens. But you don't need to know the details of how it works for this class. You just need to know everything I have just typed out here. So all of this is important. The central dogma itself is all of this. It's that DNA stores the information, and the order of nucleotides and information can eventually be translated into the order of amino acids for a protein. And the intermediate step is making an RNA copy of a gene. You can think about what's going on in your own cells, in a human cell. The DNA is in the nucleus, and that's where transcription happens. And the messenger RNA is then pushed out of the nucleus, and ribosomes are in the cytoplasm. So there's that. In bacteria, everything is kind of mixed together. And so there are lots of different reasons why this system works and why it's set up this way, and I'm happy to talk to you about them in another time. So that's enough of this video. It's a very important topic. You have to know this. You have to know the terminology that I've bolded here. And you have to know what DNA and RNA and proteins are, if only at the level of detail that I've gone into here. So I won't ask you about their structures beyond what they are made of. So you need to know what a nucleic acid is. It's a polymer of nucleotides. You need to know what a protein is. It's a polymer of amino acids. Those are the things you need to know. And with this information, what we're going to be able to do is understand how cells divide. We're going to understand how viruses work. We're going to understand how antibiotics work. And all of that really requires knowing the central dogma inside and out. So there are some slides about that, starting slide 25 in lecture 2. And I really go through the same thing. This is the translation between nucleotides in RNA. So 3 nucleotide codes in RNA encode amino acids, or they correspond to various amino acids. And this is the table. This is a version of the table that we can use to understand how cells do it. This is the genetic code, and a ribosome basically does that translation and makes the protein based on the RNA sequence. So this is the central dogma. Here's a nice picture of it. Okay, thanks for your attention to this, and I look forward to seeing you soon.